We're constantly hearing about new viral threats or sometimes old ones coming back.
Hmm.
And when something like monkeypox reappears and starts spreading globally, you have to ask, are the treatments we have actually, effective enough?
That's the critical question, isn't it?
Absolutely. So today we're doing a deep dive into one of the main antivirals being discussed, Tecovirimat, also known as TPOXX.
Right.
We're going to look at how it was developed, how it got approved, and really focus on its role against orthopoxviruses, especially monkeypox right now.
And we've got some solid sources to draw on.
Yeah, we do. There's a detailed article from PubMed Central, the original FDA press release, about its smallpox approval, a really key clinical trial report from the New England Journal of Medicine.
That one's crucial, the DRC trial.
Exactly. Plus some insights on resistance from newsmedical.net, the FDA label info, and a general overview from Wikipedia.
There's quite a bit to unpack.
Our mission here is basically to get a clear understanding of Tecovirimat, its background, how it works, what the trials really found, and sort of the nuances of how well it actually works against monkeypox. Let's get into it.
Okay, so monkeypox, or MPX, it's definitely a global threat now. It's
Yeah.
caused by the monkeypox virus, MPXV, which is related to smallpox.
Right, an orthopoxvirus.
Exactly. It was first spotted, interestingly, in lab monkeys in Denmark back in 1958. That's where the name comes from.
Ah, okay.
But the first human case wasn't actually seen until 1970 in the Democratic Republic of Congo.
So it's been around in humans for a while, mainly in certain regions.
For decades, yeah, primarily endemic in several African countries.
Hmm.
But then things changed. The first case outside Africa popped up in the U.S. in 2003.
I remember that linked to prairie dogs, wasn't it?
That's the one. And then much more recently, in 2021, 2022, we saw this pretty dramatic multi-country outbreak. Americas, Europe, Eastern Mediterranean, Western Pacific, plus ongoing spread in Africa.
And how does it spread? That seems key to understanding the outbreaks.
It is. Transmission is mainly through direct contact, bodily fluids, those characteristic skin lesions,
Okay.
respiratory droplets, though maybe less efficiently than some other viruses, and also contaminated materials like bedding or clothing.
So close contact is a major risk factor.
Definitely. Household members, sexual partners, they're at higher risk.
You mentioned it's related to smallpox, but are there different versions of monkeypox itself?
Good question. Yes, there are two main types, or clades. There's the Central African or Congo Basin clade and the West African clade.
And one's worse than the other.
Generally, yes. The Congo Basin clade is considered more virulent, causes more severe disease, and seems to spread more easily between people.
Okay.
The recent global outbreak, the one outside Africa, is thought to be mostly due to the West African clade, which tends to be milder.
And what does the infection actually look like in a person?
Well, it starts with the virus replicating, then it gets into the bloodstream, that's primary viremia, then it spreads to the lymph nodes, causing them to swell up. That's called lymphadenopathy. That's actually a key feature that helps doctors tell it apart from smallpox or chickenpox.
Ah, that's useful.
Then there's a second wave in the bloodstream, secondary viremia, and that's when you typically get the classic symptoms. Fever, chills, bad headache, muscle aches, backache, feeling really exhausted.
Sounds rough.
And then the rash appears, often starting on the face and spreading. If it's not treated, or if someone's immune system is weak, there can be serious complications.
Like what?
Secondary bacterial infections of the skin lesions, sepsis, lung issues like bronchial pneumonia, sometimes encephalitis, which is inflammation of the brain, or eye problems like corneal infections.
Yeah.
And in some cases, it can be fatal.
Wow. So given it can look like other things and the potential severity, what factors really allowed it to spread so widely recently?
It's likely a combination of things. A big one is the end of routine smallpox vaccination.
Because that gave some protection.
Exactly, some cross-protection. So now we have populations with less immunity. Also, increased contact between people and animals that might carry the virus, more international travel.
Makes sense.
Maybe some decrease in international health coordination, difficulties in diagnosing it quickly, perhaps not enough research funding historically, and definitely limited availability of specific treatments.
Which brings us back to Tecovirimat, TPOXX. Is this the ray of hope?
Well, it's certainly positioned as a key tool. Tecovirimat, or TPOXX, is what's called a small molecule drug, technically a tetracyclic acyl hydrazide.
Okay, science terms.
Yeah, basically it describes its chemical structure. It's designed specifically to target orthopoxviruses, which includes monkeypox.
And it's approved?
Yes and no, depending on where you are. The European Medicines Agency, the EMA, approved the capsule form in January 2022 specifically for monkeypox, smallpox, and cowpox.
Okay, so Europe's covered for monkeypox.
Right. But interestingly, the US FDA and Health Canada have only approved it for smallpox so far.
Smallpox. But smallpox is eradicated. Why focus on that?
Bioterrorism concerns, primarily. The worry was smallpox being used as a weapon. That's why Tecovirimat was developed, largely with U.S. government funding, and why there's a big stockpile of it in the U.S. Strategic National Stockpile.
Ah, okay. That makes sense. But if you can't test a smallpox drug on humans with smallpox, how did they prove it worked?
That's where something called the animal rule comes in, because human trials for smallpox are unethical and impossible.
Right.
The FDA allows efficacy to be shown in relevant animal models. For Tecovirimat, this meant studies in rabbits infected with rabbitpox, which is related, and crucially, in non-human primates, monkeys infected with monkeypox virus.
So they used monkeypox in monkeys as a stand-in for smallpox in humans.
Essentially, yes, particularly using a virulent strain, the Zaire 79 strain of monkeypox.
Yeah.
And those animal studies were actually quite
Oh,
promising.
what did they find?
They found that Tecovirimat provided 100% protection against death in the infected monkeys.
Wow, 100%.
Yeah, at the doses tested. It also significantly reduced the amount of virus in their bodies and the number and severity of the skin lesions. Importantly, it worked even if they started the treatment after the virus had already begun circulating in the blood.
That sounds pretty convincing, at least in animals.
It was.
Oh.
And similar good results were seen in the rabbitpox models, too. So these animal studies were really the foundation for its approval, especially under the animal rule pathway for smallpox.
Okay, so how does it actually stop the virus? What's the mechanism?
It's quite specific. Tecovirimat targets a protein called VP37.
VP37.
Yeah, and this protein is only found in orthopoxviruses. It's essential for the late stages of the virus's life cycle inside an infected cell.
How so?
Think of the virus needing to get packaged up properly before it can leave the cell and infect others. VP37 is critical for forming the outer envelope or membrane around the mature virus particle.
Like putting it in a shipping box.
Sort of, yeah. It needs this envelope to exit the cell efficiently and spread. Tecovirimat basically jams that packaging process. It inhibits VP37, so the virus can't form that crucial envelope.
So it gets stuck inside the cell.
Effectively, yes.
Yeah.
It prevents the formation of these extracellular enveloped virions, which are the form that's really important for spreading from cell to cell and throughout the body. So it stops the spread.
That's a neat mechanism, very targeted. So if someone does get prescribed tecovirimat, say for monkeypox under compassionate use or in Europe, what do they need to know? Dosage, side effects.
Right. For adults, the standard oral dose is 600 milligrams twice a day for 14 days. Pediatric doses are adjusted by weight.
Okay.
A really key point is taking the capsules with a fatty meal. This significantly boosts how much drug gets absorbed into the body.
Ah, like some other meds. Good to know. What if someone can't swallow capsules?
The contents can be mixed with milk or soft food like applesauce. Now, contraindications, reasons not to take it, there aren't any listed for the capsules. But the IV version shouldn't be used in people with severe kidney problems.
Okay. Any warnings?
Yeah, caution is advised for people with weakened immune systems, immunocompromised individuals. There's some suggestion it might be less effective for them. Also, care is needed with existing kidney or liver problems.
And side effects? What's common?
The most commonly reported ones are things like headache, some dizziness, nausea, maybe vomiting, diarrhea, abdominal discomfort. Generally considered fairly well tolerated, but those are the main ones.
Any drug interactions to watch out for?
Yes, there are known interactions. It can affect how the body processes certain other drugs, notably repaglinide, which is used for diabetes, and midazolam, a sedative. So doctors need to be aware of other medications a patient is taking.
Okay, so we have the animal data, the mechanism, the approvals. But the real test is in humans with monkeypox, right? Which leads us to that New England Journal of Medicine study in the DRC.
Exactly. This is where the story gets more complex. The PALM007 trial. It was done in the Democratic Republic of Congo, dealing with the Clade I monkeypox virus, the more severe type.
And it was a proper trial. Randomized, placebo-controlled?
Yes. Double-blind, randomized, placebo-controlled, the gold standard design. They were primarily looking at how long it took for the skin lesions to fully resolve, crust over, and fall off.
Okay, so what did they find? Did Tecovirimat speed things up?
This was the surprising part. They found, well, basically no significant difference. The median time for lesions to resolve was about 7 days in the group getting Tecovirimat and 8 days in the group getting the placebo.
Only 1 day difference and not significant.
Correct. Statistically, There was no significant difference between the two groups for that primary endpoint.
Wow, that's not what you'd expect after the animal studies.
Not at all. And they looked at other things too, like how quickly the virus became undetectable by PCR testing in skin lesions or blood. Again, the rates of clearing the virus by day 14 were pretty similar between the Tecovirimat and placebo groups.
So across the board, it didn't seem to make much difference in this trial.
In terms of speeding up recovery or clearing the virus faster, no, not significantly, according to their main measures. Now, it's worth noting the overall mortality rate in the trial was lower than what's typically reported for Clade I monkeypox in the DRC.
Okay, that's something.
But the drug itself didn't show a clear benefit over placebo for the primary outcomes measured. And the rates of adverse events were comparable between the groups, suggesting it was safe, but just maybe not effective in that context.
So why? Why would it work so well in monkeys, but seemingly not in this human trial? That's the big question, surely.
Absolutely. And the researchers and others have discussed several possibilities. One is potential resistance.
The virus evolving to beat the drug?
Possibly. The newsmedical.net piece touched on this. Maybe certain strains, perhaps more common in the DRC or specific Clade I lineages, have ways to get around Tecovirimat's mechanism. That needs more investigation.
Okay, what else?
Another major point that came up, not just in this PALM trial, but also in another observational study called STOMP, was that the levels of Tecovirimat measured in the blood of some patients were lower than expected.
They weren't absorbing it well enough, even with the fatty meal advice.
It seems that way for some. Potential reasons could include malnutrition, which is sadly common in that region, maybe other coexisting infections interfering with absorption or metabolism, possibly interactions with other drugs or traditional medicines they didn't capture, or even genetic differences in how people process the drug.
So maybe they just weren't getting enough of the drug into their system to work effectively.
That's a strong possibility being considered. Though interestingly, in the PALM trial, even when they looked at the drug concentrations they did measure, those levels didn't clearly correlate with faster lesion healing or virus clearance anyway.
Huh. So it's still a bit of a puzzle.
It really is. It suggests the relationship between drug level, virus strain, and patient outcome might be more complicated than initially thought.
So despite being approved for monkeypox in Europe, this trial really throws a spanner in the works regarding its effectiveness, at least for Clade I.
It certainly does. It underscores that translating promising animal data into clear human benefit isn't always straightforward, especially with potentially diverse viral strains in patient populations. You have to be cautious about assuming broad effectiveness.
Now, something else mentioned in the sources, particularly PubMed Central, was the patent situation around Tecovirimat. Why is that relevant here?
Patents are always relevant with drugs. SIGA Technologies holds a pretty extensive portfolio of patents covering Tecovirimat.
Not just the molecule itself.
No, it covers different forms of the drug, like specific crystalline structures, the actual pharmaceutical compositions, how it's made into a pill, or liquid methods of using it for treatment, liquid formulations, even the manufacturing processes.
So they have it pretty locked down.
Largely, yes. Though there are also some patents from other groups mentioned, looking at things like injectable versions or potential combination therapies. There was one suggesting combining it with cidofovir, another antiviral.
And why does this matter for, say, global access or cost?
Well, strong patent protection can influence the price of a drug, who can manufacture it, and how easily it can be made available, especially in lower-income countries that might be heavily affected by outbreaks. It's a key factor in the background.
Okay, so pulling all this together, where does this leave us with Tecovirimat? It started as this potential biodefense drug, showed promise in animals, got approvals, but then this major human trial raises big questions.
That sums it up pretty well. It's a significant antiviral in the Orthopoxvirus space, but its story is complex. The approval status varies, and the efficacy data, especially from the DRC trial with Clade I MPXV, introduces real uncertainty.
So not quite the straightforward solution we might have hoped for?
Perhaps not universally. It highlights the critical need for ongoing research. We need to understand optimal dosing better. Maybe higher doses or different formulations are needed. We need to seriously investigate combination therapies. Maybe Tecovirimat works better alongside another drug, especially if resistance is a factor.
Like that cidofovir combination idea?
Potentially, or others.
Yeah.
In vitro lab tests have hinted at combinations being useful. And crucially, we need continued monitoring of patients who do receive Tecovirimat, especially vulnerable groups like immunocompromised individuals, to see if there are benefits or side effects we haven't fully captured yet.
So Back to our initial question, how confident can we be in our treatments when viruses like monkeypox reemerge? It sounds like the answer, for Tecovirimat at least, is it's complicated.
It is complicated. We
Mm,
have a tool that seemed very promising based on initial data and its mechanism.
right.
But real-world clinical evidence in humans, particularly against the more severe clade in an endemic region, paints a much more nuanced picture. It really drives home that fighting infectious diseases requires constant learning, research, and adaptation.
It absolutely does. Which leaves us with a final thought, maybe something for our listeners to chew on.
Okay.
Given that viruses naturally evolve and drug resistance is always a potential threat, how should global health strategies really prepare for the next outbreak?
That's the billion-dollar question, isn't
It
it?
feels like it. How do we balance that urgent need to deploy countermeasures quickly when something flares up versus the equally vital need for rigorous testing to make absolutely sure they work effectively and safely in all the different people and places they might be needed?
Finding that balance between speed and certainty, that's a huge challenge. It requires robust surveillance, flexible trial platforms, international cooperation, and maybe rethinking how we develop and stockpile these countermeasures in the first place.
Definitely a lot to think about.